Tardigrade from 16 million years ago sunk in amber. Tardigrades, also known as water bears, are a diverse group of unusual, microscopic invertebrates. They are known primarily because they can survive in extreme conditions. In 2007, they survived a trip to space, and after returning from the vacuum, they were still able to reproduce. These amazing animals are found on all continents and in different environments. Now scientists have published a description of a tardigrade found in amber from 16 million years ago. Scientists know that tardigrades lived millions of years ago and survived all the mass extinctions that affected our planet. However, despite their long evolutionary history and global distribution, the fossil record of water bears is extremely sparse. All because of their microscopic size and body that does not undergo mineralization. All the more remarkable is the discovery of a new species of tardigrade from millions of years ago. The research results were published in the journal Proceedings of the Royal Society B. Researchers used the most modern techniques to determine important anatomical features of the animal embedded in amber. Thanks to this, they determined that so far no similar tardigrade has been found. The new fossil was named Paradoriforibius chronicarabius. This is only the third tardigrade found in amber described by scientists. The former two are named Milnesium swolensky and Bjornlegi, both found in Cretaceous North American amber. Paradoriforibius chronicarabius is the first fossil of this species from the Miocene. An individual that lived about 16 million years ago. The authors of the research are Philip Barden from the New Jersey Institute of Technology and Marco A. Mapalo and Professor Javier Ortega Hernandez from the Department of Biology at Harvard University. The difficulty in working with this specimen was that it is so small. We needed a special microscope to fully see the inside of the fossil, Mapalo explains. The light emitted by special devices for similar analyses is well suited for studying the morphology of insects, such as spiders or flies. However, the total body length of Paradoriforibius chronicarabius is only 559 micrometers, or just over half a millimeter. With such a small specimen, the microscopes normally used would not reveal all its secrets. The epidermis of tardigrades consists mainly of chitin, which is fluorescent and easily excited by lasers. By using the phenomenon of fluorescence, the insides of the fossil can be better visualized. The new research method allowed the detection of claws and structures in the animal's throat and intestine. Although the outside of the microscopic invertebrate looked like its modern cousins, the insides were different from the currently known species. This justifies the creation of a new genus within the existing group of tardigrades, says Mapalo. Paradoriforibius chronicarabius is the only genus that has such a specific, unique set of features in the superfamily Isohypsibioidea, explains the researcher. Tardigrade fossils are rare, says Ortega Hernandez. With our new study, the complete listing includes only 4 specimens, of which only 3 are formally described and named, including Paradoriforibius chronicarabius. So our work represents basically 1/3 of the fossil record of water bears known so far, the scientist emphasizes. The authors note that tardigrade fossils in amber are well preserved due to their small size and habitat preferences. For this reason, amber deposits are the best source of obtaining new specimens from millions of years ago. However, this does not mean that finding these microscopic invertebrates is easy, as it requires excellent observational skills and specialized knowledge. We know that tardigrades are closely related to arthropods and that their rapid development occurred during the explosion of life forms in the Cambrian. The problem is that we have so little fossil evidence for their development, says Ortega Hernandez. If we look at the external morphology of the tardigrades, it can be assumed that there have been no changes in their organisms, adds Mapalo. However, using modern technology, we saw new features that are not found in modern species. This helps us understand what changes have occurred in their organisms over millions of years, argues the researcher. Scientists are planning further research on fossil tardigrades. People all over the world like the same fragrances. Researchers have determined the most pleasant of them. Scientists say that pleasant and unpleasant odors are common to people from different cultures. This may point to the evolutionary basis of our seemingly universal scent preferences. In the new study, researchers asked people from 10 different cultural groups, including indigenous hunter-gatherers, rural and urban people, to smell 10 unique scents and rank them from prettiest to least pleasant. We wanted to see if people around the world perceive smells in a similar way and like the same smells. Or if it's a cultural acquisition, says neuroscientist Arten Arshamian of the Karolinska Institute in Sweden. It's traditionally thought to be a cultural issue. But our research shows that culture has very little to do with it. The research results were published in the journal Current Biology. Scientists have traveled very far, collecting data in many cultural environments, representing different lifestyles. They were, among others, in Thailand, Mexico, Ecuador and the United States. They studied people living in deserts, tropical rainforests, high mountains, coastal regions, and more. Participants in the study included groups of hunter-gatherers, including Samak Berry from the Malay Peninsula in Thailand. Several farming communities, including Chachi from Ecuador, and people from New York, representing a contemporary, racially and ethnically diverse environment urban. In total, over 280 people from all over the world were tested. Participants in the study were given 10 scented devices. They were asked to smell each and order them in a row from most pleasant to most unpleasant. Vanillin, the main component of vanilla extract, turned out to be the most pleasant smell, followed by ethyl butyrate, fruit-scented, often used as a flavor enhancer in fruit-flavored foods, resembling the smell of peaches. In third place was linalool, which has a floral scent. The least popular was isovaleric acid, which is known for its pungent, unpleasant odor, associated with cheese, soy milk and the sweat of human feet. Similar studies have been carried out before, but only the most recent ones have put such great emphasis on cultural diversity. Researchers expected that people's assessment of odor pleasure reflected a combination of cultural tradition, personal sensitivities, and universal preferences, but the extent of each of these influences has been unclear until now. Arshamian's team found that the influence of cultural traditions plays only a minor role in people's scent preferences. Fragrance enjoyment rankings are strongly correlated across all cultures studied, when people rated individual scents in terms of perceived pleasure. Participants' culture only explained about 6% of the smells variance, while 54% was due to individual preferences, and the molecular profile of the chemical substance of the fragrance explained about 40%. In other words, while people can and do evaluate different scents differently, most of the observed differences seem to be a matter of personal preference rather than a reflection of culture. There was significant global consistency. Altogether, this shows that human olfactory perception is strongly constrained by universal principles, the researchers explain in the paper. In other words, while people can and do evaluate different scents differently, most of the observed differences seem to be a matter of personal preference rather than a reflection of culture. There was significant global consistency. Altogether, this shows that human olfactory perception is strongly constrained by universal principles, the researchers explain in the paper. Why does the perception of odors seem to be at least somewhat universal across cultures and is based on the physiochemical properties of odorous substances? There is a strong possibility that a preference for certain smells may have served an evolutionary purpose, somehow increasing our chances of surviving in long-forgotten times. If so, the study results could help us explore this possibility more closely, the researchers suggest. We now know that there is a universal perception of smell that is driven by the molecular structure of the fragrance compounds. This explains why we like or dislike a particular fragrance, says Arshamian. The next step will be to investigate why this happens by linking this knowledge to what happens in the brain when we smell a particular smell.